Hello, my name is Alba Dumawala, and today I'll be presenting my final genetics project on the genetic mapping of three X-linked mutations, white, miniature, and sable in Drosophila melanogaster. This work was completed using recombination data from a three-point test cross, and the data were combined with partner groups to increase accuracy. So why we use Drosophila melanogaster? We use Drosophila melanogaster because it's a well-known and classical model organism in genetics. And this is because it has a short life cycle and visible mutant phenotypes. The X-linked traits are especially informative because males are hemizygous for the X chromosome, meaning their phenotype directly reflects the allele they inherit. And the recombination frequency between linked genes allow us to estimate their relative distance along a chromosome. This experiment focused on three recessive X-linked mutations: white, which affects eye color; miniature, which affects wing size; and sable, which affects body pigmentation. All three genes are located on the X chromosome, and their physical proximity suggests genetic linkage. Genetic distance between these genes are measured in map units, also known as centimorgans. The objective of this study was to analyze recombination data from an X-linked three-point test cross in Drosophila melanogaster to determine the gene order and genetic map distances among the white, miniature, and sable loci, and to evaluate crossover behavior through interference analysis. The null hypothesis states that there is no significant difference between the observed and expected recombination frequencies, and that any deviation from expected values is due to random chance. For the experimental design, we use FlyLab to simulate the test cross. So we use mutant virgin females that were crossed with wild type males to generate F1 females that were heterozygous for all three genes. The recombination occurs only in females, making these F1 females essential for detecting crossover events. The F1 heterozygous females were then test crossed with mutant males because the males contribute only mutant alleles. The phenotypes of the offspring directly reflect the gametes produced by the females. A total of 8 phenotypic classes were expected. The offspring were categorized into 8 phenotypic classes, representing parental types, single crossovers, and double crossovers. To improve accuracy and reduce sampling error, data from my experiment were pooled with data from partner groups, increasing the total number of offspring analyzed. The parental versus recombinant gametes. The most frequent phenotypic classes were identified as parental, indicating two no recombination, and the intermediate frequency classes represented single crossover events, and while the least frequent classes represent double crossovers. Gene order was determined by comparing parental phenotypes with double crossover phenotypes. In a double crossover, only the middle gene differs from the parental arrangement. This comparison identified miniature as the middle gene, establishing the gene order as white, miniature, and sable. For the map distance calculations, we use recombination frequencies and using the pool data. The distance between white and miniature was calculated as 25.92 map units, which rounds to 26 map units, and the distance between miniature and sable was 7.7 map units, which rounds to 8 map units. These values are consistent with classical genetic mapping expectations. To analyze interference, the expected number of double crossover was calculated from the product of the two single crossovers frequencies, and the coefficient of coincidence was calculated as the observed number of double crossovers divided by the expected number, giving a value of 0.98. Um, Interference was then calculated as 1 minus the coefficient of coincidence, resulting in an interference value of 0.02. This indicates extremely low interference, meaning crossovers events in this region occur almost independently. Also, a chi-square test was performed to compare observed and expected double crossovers frequencies. The chi-square value obtained was 0.013 with 1 degree of freedom. This value is far below the critical value of 3.841 at p equals 0.05. So we fail to reject the null hypothesis. That means that we accept the hypothesis. This indicates that the observed data closely match the expected values and that any deviation is likely due to random chance. In conclusion, this experiment successfully mapped three X-linked genes in Drosophila melanogaster. The gene order was determined as Y miniature Sabel, and genetic distances were calculated using recombination frequency. The pool data revealed very low interference and a strong agreement between observed and expected values, supporting classical recombination theory. Overall, this study demonstrates how three-pointer crosses and recombination analysis can be used to understand gene linkage and chromosome structures. Thank you for watching.